Next is intraembryonic, intracranial, and intravenous inoculation. These are very rarely used inoculations. However, sometimes we have to use these inoculations. So, inoculated intraembryonically or intracranially and then incubated for a few days and see the results whether the viruses have been grown or not. And lesions are two types of the lesions we can see, or the death is the observed, and in this way we can see that viruses are inoculated and viruses can be isolated from different specifically from dead embryonated eggs. These inoculations are not widely used, as we have already studied. These are very rarely and not used widely. And the embryonic inoculations, the embryos are inoculated and incubated for a period of 1 to 6 days. So in this way we can inoculate the virus embryos into 1 to 6 days, and they are observed twice, at the morning and at the evening, whether the death of the embryo occurs or not. And some viruses such as equine encephalomyelitis virus will kill the embryo within the 24 hours. Similarly, some other viruses will not be able to kill the viruses so quickly, and it will be take maybe about 2 days or 3 days. And some of the viruses readily multiply within the embryonic tissue, and some are seldom very lethal and does not multiply in the embryonic tissue. So therefore these all things are dependent on the viruses, and sometimes their embryonic death occurs. Therefore it will be observed from 1 to 6 days whether what kind of the lesions are observed and the death of the embryo occurred or not. Similarly, if the death of the embryo occurs, then there will be the collapse of large blood vessels of the choriodontoses, so that they are difficult to see upon the candling of the eggs. Most of the time we observe the lesions by the candling of the eggs. However, in case of the death of the embryo, it will be not possible. We cannot see, so they are quite distant while the embryos are alive. Normal movements after embryos are no longer observed if the embryo is morbid or death. So therefore it will be very easily that we can compare between the death of the embryo or with the live embryo. If the death embryo will be occur, the large vessels will be collapsed and it will be we cannot see the choriodontoses, while the normal movements of the embryo are no longer observed in the death of the embryo. Similarly, if the embryo is not dead even after the 5 or 6 days of the incubation, it is killed experimentally by ourselves, and you can see and examine the lesions after killing of the embryo by ourselves. We can see a little video here also to see most interestingly how the intraembryonic or intracranial and intravenous inoculations are occurring in the body.